We will study Riccia, its sporophytic phase. This phase in the life cycle is a direct result of sexual process. It comprises the zygote, embryo and the sporogonium. Zygote, it is the fusion cell formed by the union of the sperm with the egg and the pioneer structure of the sporophytic phase. It secretes a wall around it and enlarges in size. The zygote is retained within the venter where it begins to grow immediately. It enters upon no resting period. The zygote differ from the unfertilized egg in the following two respects. It is a diploid. It has a diploid nucleus. It has a cellulose cell wall around it. The egg before syngamy has a haploid nucleus and is naked. Development of embryo. The zygote lying in the venter undergoes repeated cell division and cell enlargement. A spherical mass of undifferentiated cell called an embryo is formed. It fills the venter cavity. The archegonium is persistent. The venter expands as a closed envelope over the developing embryo. It becomes two-layer thick and is known as calyptra. The neck of the archegonium later withers. The first division of the zygote is by a nearly horizontal wall. It is in a plane of right angle to the long axis of the archegonium. The next division is at right angle to the first. The young embryo now consists of 4 cells. It is the quadrate stage of the embryo. All the 4 quadrants are nearly equal. Each quadrant, as a rule, divided by vertical wall at right angle to the preceding one. The embryo at this stage consists of 8 nearly equal cells. Only 4 are seen in longitudinal section. It is the octave stage. Gerber and Lewis reported the formation of 4 cell filament instead of a quadrate type of embryo in some species of Riccia. Succeeding division is the 8 cell embryo are in an irregular sequence. A more or less spherical mass of 20 to 40 undifferentiated colorless cell is produced. See, young sporogonium, each of the outer cell of the undifferentiated spherical embryo divided by periclinal wall. A single layer of outer cell is separated from an inner central mass of cell. The former is called amphithecium and the later endothecium. The cells of the amphithecium are large and flat. They divide by anticlinal wall to form the protective sterile jacket layer which remain one cell thick and constitute the wall of young sporogonium. The cells of the endothecium are all alike. They constitute the first cell generation of the sporogenous tissue. It is called the archisporium. As the sporogonium advances towards maturity, the archisporial cell divide and redivide to form a mass of sporogenous cell. The cell of the last cell generation of the sporogenous mass function as a potential spore mother cell or the sporocyte. The spore mother cell are the last structure of the sporophytic phase. During further development of the sporogonium, the sporocyte begins to separate and become more or less rounded. Their wall become mucilaginous and the content densely granular. Practically all the sporocytes divide meiotically to form spore. A few, however, are said to disintegrate to form a nutritive fluid and are called the nurse cell. The nurse cell, as they are the forerunner of the elaters of the most advanced member of the Marchians. As the spore mother cell enter the meiotic division, their wall disintegrate. The wall of the sporogonium and the cell of the inner layer of calyptra also disintegrate. The round spore mother cell lie free in the cavity of the sporogonium, surrounded by an outer layer of calyptra. The developing spores are bathed and nourished by the nutritive fluid formed by the disintegration of the wall of the spore mother cell. Non-functional spore mother cell, jacket layer and the inner layer of the two cell calyptra. Thank you.